My name is Rachel Baum, and Baum and Nguyen and I will be describing our JCB paper, A Truncated Isoform of Connexin43 Caps Actin to Organize Forward Delivery of Full-Length Connexin43. In this manuscript, we study Connexin43 gap junctions to understand the overarching question of delivery specificity of membrane proteins. Connexin43 ion trafficking to particular regions of the cell membrane is crucial for the formation of gap junction plaques, which allow for intracellular communication and are vital to cell survival, and in the heart, function to coordinate each heartbeat. Forward trafficking of Connexin43 undergoes a highly regulated process called targeted delivery, whereby newly synthesized Connexin43 hemichannels are packaged into vesicles and trafficked on microtubules to specific cadherin-rich membrane subdomains. Targeted delivery relies on microtubules as trafficking highways, and actin is the key to stabilize the forward delivery system. GJA1 mRNA, which encodes Connexin43, does not undergo splicing, however produces isoforms of Connexin43 via internal translation. This occurs when internal methionines, downstream of the start codon, serve as translational initiation sites, and peptides are formed that are effectively N-terminus truncated isoforms. The 20 kilodalton isoform, GJA120K, has previously been discovered to be crucial for Connexin43 trafficking and has also been associated with organizing the cytoskeleton, and we sought to find the precise link between its role in trafficking and its interaction with cytoskeletal proteins. We found that overexpression of GJA120K results in reorganization of the actin cytoskeleton. When GJA120K is overexpressed, we see thicker actin fibers as well as a population of circular actin puncta within the cytosol of the cell. When we quantified the number of actin puncta per cell, we found a large increase in cells expressing the smaller GJA120K isoform. Cells expressing only full-length Connexin43 protein required additional GJA120K to rescue the puncta phenotype. In order to test that GJA120K directly binds to actin to induce the puncta phenotype, we utilized the cell-free actin polymerization assay, where we were able to use turf microscopy to image actin polymerization in the presence of purified GJA120K. We found that GJA120K inhibits polymerization in a dose-dependent manner similar to that of a class of actin-modifying proteins known as actin-capping proteins.
The actin puncta phenotype is similar to the previously reported actin rest stop structures at which Connexin43 vesicles localize and are necessary for microtubule-based Connexin43 trafficking to the cell membrane. We asked whether GJA120K capped actin could regulate microtubule dynamics. We performed time-lapse imaging to track the trajectories of EB1, which occurs at the plus end tips of growing microtubules, and found that actin puncta induced by GJA120K coincides with the occurrence of microtubule branching, and that their presence increases microtubule tract overlay. These data suggest that GJA120K organizes actin puncta as a site of actin microtubule interactions. We ask if the binding of GJA120K and actin is crucial for directing microtubule-based Connexin43 targeting to cell-cell borders. We deleted the GJA120K terminal domain responsible for actin binding and found that this deletion reduces Connexin43 trafficking to the cell borders. Importantly, this is rescued by the reintroduction of exogenous full-length GJA120K. Finally, we ask if the microtubule-based delivery of Connexin43, which is organized by GJA120K and actin interaction, modulates Connexin43 gap junction function. We knock down GJA1 gene and exogenously introduce back GJA120K and Connexin43 to explore gap junction permeability. Consistent with increased Connexin43 delivery to cell borders, exogenous GJA120K and Connexin43 improve gap junction permeability, as evidenced by increased spreading of gap junction permeable dye. In summary, we discovered that GJA120K, a smaller isoform of the full-length Connexin43 ion channel gap junctions formed by internal translation from the same GJA1 mRNA, caps actin to form actin puncta necessary for organizing microtubule-based trafficking pathways. These in turn enhance the cytoskeleton orientation towards cell-cell borders and the delivery efficiency of gap junction channels to their designated location at the cell membrane. In short, the mRNA of a full-length membrane protein encodes a smaller isoform that physically organizes the cytoskeleton delivery apparatus. Thank you.